Okay, so welcome everyone to the second Moonshot Symposium. My name is Thomas Hermans. I'm the program manager, and the agenda for today is a short introduction. We're just waiting for Sir Peter Ratcliffe to figure out some connection issues. So while that's going on, I will quickly continue with a very brief introduction about the challenge. So the origins of the JEDI project are to quickly identify molecules that do something against coronavirus. As you all know, this is a huge problem. And the classical drug discovery process, as you know, takes a lot of time, has high cost. And there are some interesting things going on, of course, worldwide, like the huge programs like Solidarity, Discovery and Recovery, that we hope also Professor Ratcliffe will be able to say something about. But still, we can probably do more, and we can probably do better as a community worldwide to tackle this challenge. So the big question is, can we disrupt drug discovery to tackle this crisis and also future pandemics? So how can we do things much faster and much better than what we've been doing so far? That's the big question, and we hope we can contribute to that. So if you look at the typical drug discovery pipeline, a lot of effort and money is spent on actually finding an appropriate target, for example, a protein target, and generating the first hit compounds. So this is the stage that we're trying to tackle. And of course, that's followed by a long phase of development, safety checking and FDA approval, which is a bit beyond our scope at the moment. But what the proposition is that the JEDI is trying to take is to basically use collective intelligence of the teams, right? So the teams collectively, and we're now over 100, as André will explain you later, we're over 100 teams that actually together decided what is the best protein targets in this case, also what to include in the compound libraries. So really screening through billions of compounds. And we combine this with high throughput screening. And we hope that this leads to significant benefits. So the first year in traditional drug development, which is the generation of finding a target and generating a hit, is typically 1 year. We're trying to do this in 4 months. Also, the cost saved, we're trying to do all of these steps with, let's say, €500,000 versus the traditional cost, which is closer to 21 million euros, as from this Nature Reviews paper on drug discovery. So the big challenge we have is how to screen billions of compounds with a potential blocking interaction versus the virus. And we have set up these methods. So first of all, we want to screen a huge chemical space. So each team has to screen more than 1 billion compounds. There are 100 teams. So at least we can be sure that there are more than 5 billion unique chemical compounds and maybe even more. We encourage teams to use different methods. So we can think about docking, molecular dynamics, other machine learning approaches, and combine that with the power of high performance computing. And then the unique approach of the JEDI challenge is that when we have all the information from all the teams, via a cluster analysis, we will see which regions in chemical space are actually densely populated. And actually, we will pick from those spaces to find the best compounds. And lastly, there's experimental validation through biophysical assays, live virus testing, and animal models. Okay. So André, please update us on the progress so far.
Yeah, so thank you everybody to attend, and thank you for the team and the many people who helped put together this second Moonshot Symposium. So we're very happy to see the number of teams who are participating. You are now 124 from all over the world, 40% from Europe, 30% from the Americas, Asia, and Africa. We estimate around 500 scientists are involved. At this stage, we estimate, because obviously we don't have the final numbers, the stage 1 of the challenge is still ongoing, is that we have already collectively screened more than 50 billion molecules. So this is an outstanding achievement, and we have still 10 days to go until July 17. As Thomas mentioned, many methods are used, and this will be very important for the cross-validation of the lead compounds that will come out of this exercise. And just also as an interesting data point, enormous amount of computing power has been used. We estimate approximately 10 million core hours have been dedicated to this challenge alone so far. So I think now I'll, without further ado, we will start with a Q&A with Sir Peter. Thomas, do you know if he has been able to connect with us? Wonderful. So welcome and thank you very much for joining us.
Okay.
So welcome, Professor Ratcliffe, first of all. For those that don't know, Sir Peter Ratcliffe, he's the 2019 Nobel laureate in medicine. He's the director of clinical research at the Francis Crick Institute. So with further ado, let's start with the first question. So maybe I can kick off. So there's a strong response to COVID-19 now, but do you think after Zika, Ebola, SARS, MERS, we now finally learned our lesson? That's the big question. And let's say to ask also the audience about that, we can do a little poll. So the question also to the audience, do you think we will be ready for the next pandemic? So please go ahead and submit your votes. I'll give it a couple of seconds. Okay, I think we are reaching a consensus here, so let's look at it. So, right, it's not looking too great according to the audience. So what do you think, Professor Ratcliffe?
I think the audience is correct. I don't think we've learned them yet. This is, I think, a wake-up call. There have been a few close shaves, and the people I know in infectious disease point this out, as you set out. How many times does the community need to be told that infectious disease remains a problem, in particular viral infectious disease remains a big problem? I guess the question that you'd like to introduce is exactly what preparedness means. It's one thing defining that we haven't done it. It's another thing, I guess, defining what we should do and what lessons there are to be learned. And I guess that's what we should debate. There are all sorts of levels to that. How do you... What sort of antiviral or vaccine or immunological or chemical entities would we like to create, and how do we smooth the platform that we could do that reasonably quickly, and how do we get what advanced warning we might be able to get of viruses that could do us damage. As you'll be aware, a lot of this has been thought through with flu, which has classically been the pandemic threat, and one that institutions like the World Influenza Center, which we have at the Crick, tries to keep ahead of. But I think I'd like to introduce and hear back what are the generic approaches that we might consider? And André and I discussed on the telephone the other week the possibility that there might be generic associations, at least to RNA viruses of this type, through oligonucleotide therapy. That would be something to think about. Instead of piling up kinase inhibitors for potential use in cancer, should we be looking at viral enzymatic targets, and how would we obtain the appropriate incentives for pharma who would undoubtedly have to be involved in advance of there being a clear market? That's something society would have to consider. And I think there are organizational problems. One you'll probably be aware of, that actually it's very difficult to get academic institutions and individual academics to pull together in things. And we've seen, at least in this country, quite a lot of disparate attempts to define a cohort, to use that for immunological studies, to use it for severity studies, and then find someone else picks a different cohort for a different purpose. Of course, there's massively more advantage in making all of the same assessments on the same cohort that you can describe the cross-correlation. But that's an organizational issue that is not trivial. It certainly could be addressed. One of the experiences that we've had, which is interesting, a colleague, Peter Robbins, suggested the use of olmetrine. This is a drug made by a French company, Servier, to improve ventilation perfusion matching in both acute and chronic pulmonary disease. This is a really interesting thing because clinically, not only might it work, it probably will work to a degree. And secondly, you could organize a clinical trial that would read out within hours, i.e. if a person changes their arterial saturation in response to drug, you would know immediately. And that's one of the problems with medicine. How do you conduct a trial with and know when you've got an effective medicine? Here we would know it straight away. The drug has been made. It was taken off the market for the original indications. So the stocks were not high. And essentially, we wanted to remake this compound, bring it up to clinical grade and use it in a clinical trial. And despite the best efforts of Peter and myself, bringing influence to bear on various institutions, we have not yet succeeded in doing that. So we'll miss the first wave, and that sort of thing really needs to be ironed out, that it ought to be possible to move more quickly with a compound that exists when you actually know the measurement you want to make. And I think that we will look forward, I hope, to some sort of rationalization of legislation in this area.
Do you think one option would be to make a quicker connection to WHO? It seems that once those compounds get highlighted by WHO, that things move maybe more rapidly. Or what's your opinion on that?
It really is a question of hierarchy of power. I have to say, this is an enormous sadness for the UK to be leaving the European Union for all sorts of reasons, but one of them is that we will no longer have necessarily one unifying regulatory body. Even if we agree coherence, it still will be not one body. And the problem, one problem that is brought into sharp focus now is we have economic connections, we have economic interdependency, we have manufacturing interdependency, but we don't have political interdependency. We don't have it at a top level political consideration, and we don't have it in relation to a number of these legislative bodies. So what we might achieve in one country will not be applicable to another. And of course, if we were to trial this agent now, we would not be able to trial it in the UK or Europe because the case incidence is low. We would need to go to South America or the US or South Asia. And that, of course, will bring its own regulatory problems. So I think it is an international issue.
So maybe on the points you mentioned before about academics sticking together or collaborating. So how can we bundle and use the collective intelligence of scientists in the world, to come up with original approaches or accelerate the development of therapeutics? Is there some kind of platform that could be imagined to catalyze this?
Well, we've sort of been caught out a little bit with the structures of the funding bodies who have often got people with great organizational capacity, but these people don't usually take a scientific line and often are not qualified to do so. So we have this slightly odd situation where everyone's urging coherence. But of course, that's one thing to urge it in general. It does need to be scientifically informed coherence. And I don't really think we've got that sort of body. You know, for instance, in industry has algorithms for leading large numbers of people. Academia doesn't. It relies on passion, as I put it in different contexts, of the individual to move things forward, which I still believe is an enormously important and powerful principle. But here I think we need some coordination. So that there's a flip side to every argument. And on this occasion, we're absent that scientific overview and administrative capacity that could bring us all together and say in what precise scientific ways we should be collaborating.
Right, but in a recent ERC interview, you wrote that it's important to gain knowledge in an unbiased way, and I always compare academics to a herd of cats, so they all do whatever they want. So in this case,
I didn't use the word herd of cats, but you're right. I did make that point, and as I say, I still believe it, that the majority of knowledge creation, of good knowledge creation, comes from the passion of scientists. It's not a good idea to impinge on that passion, and by and large, that served the world well. And to be clear, it served the world well in terms of COVID-19. We know how to conduct the polymerase chain reaction for diagnosis. We know what an antibody is. We know many aspects of viral biology which are generic. We can consider what I've just mentioned, oligonucleotide therapy, and all of that information was gained without any knowledge. And I still believe that should remain the large province of academia. That should be 80% of what we do. But now we face a slightly different challenge, and there is a case for adding to that with a different algorithm. But I really do not think we should reform the whole of academia around COVID-19. That may be ineffective, and the next threat will come from a different direction. It is one of coordination, all sorts of things, and not just. A lot of this is, one concern is undue emphasis on protection of the individual. So we have enormous problem in data coherence because it's considered that individuals did not give consent for the use of data in different ways. The large majority of people who give consent to medical research do it in a broadly generic way. They're not concerned whether the information is used in France or Germany or the UK or a broad group or a narrow group of individuals. And in my view, it's quite wrong that restraints are placed on that.
Right. So the maybe a bit more. So one thing I was thinking, like, could we make a kind of, like in the military, you have the reserve forces that just basically do their normal job under peacetime, and then when it's needed, they are called up to, all hands on deck to fight the enemy, let's say. So could we imagine something like that for scientists?
Possible, although one might hope that there are people with the talents that would come to the fore. I'll try to illustrate this, if I may, with a neutral party, which for this meeting is UK government. right? We have a liberal, it's a conservative government. By and large, though, Europe has liberal government. They are responsive to the will of the people in broad outline. They have few individuals with operational capacity. And this has been particularly exemplified in the UK, where the response has been frankly poor, poorly coordinated, poorly thought through. And that's because the people in government do not actually have the technical skills or the operational skills and are not managers in the usual sense of the word. Now, the UK would have prided itself on an excellent civil service, which would translate the ministerial flavour into something that might work. And sadly, that has not happened properly, and that's something that this country will have to consider as it goes forward. It is rather the same thing in science, that we would need that capacity in reserve, that some people would be able to rapidly understand and have the power to cut across limitations on data sharing. That will be a straightforward thing. It doesn't mean that we're embracing a top-down approach in every way. It means that we're responding to an international crisis with a change in regulation. And that has to be done by people who are, of course, able to consider changes to regulation and all the implications that might have. That's the sort of thing that I think we need.
And let's say on the more practical side, if we look at the drug discovery process, do you see any stages that we can skip or greatly accelerate to come up with new compounds, you know, within 1 year or so? I know that in our first conversations, you said that, you know, everything is on repurposing, but it seems more and more that repurposing alone will not get us to where we need to be.
Well, I think I've really given two examples which exemplify that. One, there are a class of scientific endeavor, we could call it preparedness, that could be done. Understanding the immunology that would be protective so that we can make the right type of viral vectors or viral delivery systems. As I've said, oligonucleotide therapy might be generic. So there are interventions that could be brought more rapidly to the clinic than a conventional small molecule. On small molecules, I don't easily see how the stages can be left out. I think they are all necessary. We still have to be safe and we still have to be effective. I did give you an example of something which you might imagine would be straightforward, that the compound exists. Published efficacy in other situations is already there, and yet we have problems with bringing it to a clinical trial. And I think that sort of thing could be foreshortened very greatly.
Right. Okay. And what about, so, I mean, there are, of course, a number of compounds that do something, you know, significant, like remdesivir, even though we in Europe will probably not see any of that, and dexamethasone, which seems to work for certain patients. But do you think it's already time to start making kind of cocktails, to start blending things together to get, let's say, a unified response against different, well, the one is a viral, you know, antiviral, and the other is a, you know, an immune response drug. So do you think we should, you know, maybe jump faster to?
I don't think that clinical medicine actually is quite used to this, giving different treatments to the same patient in the light of the evolution of their disease. And I think that that algorithm is quite fit for purpose. It's much easier to understand the role of dexamethasone when it's given in a controlled situation rather than as a cocktail. So I think the short answer to that is, at the moment, no. I think at the moment what we need is means of assessment of a single agent, and then the clinicians can think how to combine them most effectively for a particular patient.
Okay. So maybe to turn a bit back to the political aspect. So what do you think we can do to make the politicians listen more to scientists? I mean, or are we not positioning ourselves as scientists in the correct way? You know, should we run for politics, or what can we as scientists do to improve this?
Difficult to know where to start, really. I've been a little bit surprised by the difficulty in getting rather simple pieces of advice across. As I've said, I do think that what in the UK would be called the civil service is important. That is scientifically informed, and you have people there who are sufficiently cognizant of science that they could rapidly respond to specialist input. There is a problem in organising, in convincing a populist government to take technical advice from scientists. It's almost a contradiction in terms, and that's what we've seen exemplified in this. And I think I would say that if you're going to have a populist government, and that's what happens with elections, you have to have a technically able civil service behind it. And that means having good scientists who are receptive and open-minded to ideas as they evolve.
Okay, that's clear. André, do you have a question?
Yes, Sir Peter. I mean, I don't know if you will be able to be with us until the end of the conference. You told me that you had a relatively limited time, but at the end of this this symposium, we wanted to give some thoughts about an initiative that we want to take. I mean, building on the number of teams that have joined this this challenge, on the number of people who have said similar things than you about preparedness. We will try to focus both on identifying these basic generic steps that you mentioned, but also try to have some kind of a platform where the voice can be heard, because the more prescriptive we are, the less effective we will be for the next pandemic, which will be different than this one. Would you could you give us some advice about how this kind of a coalition built on science, but really with an impact, clear societal impact in mind, how would you see this happening? How can we have most impact with the work that we are doing on this specific preclinical drug discovery process?
Well, I can't give too specific a set of pieces of advice, André. I think we have to be very realistic that those things that we put forward have a pretty good chance of medical efficacy. I would put, for instance, oligonucleotide therapy in that it's not proven, but there are examples. So the concept is there, and it's definitely scalable. I would put understanding the type of immune response which is restraining here. These are very tangible issues that can be focused and have a real output that academia can deliver. There is a problem in academia saying, well, we'll just do everything. In the case of my own institution, will it make sufficient number of inventions to solve any economic problem accruing from Brexit? Well, that's not, you might promise that it'd be popular, but you won't achieve it. So I think academia has to gain confidence by being able to deliver what it says. So really that we should concentrate on things where we're reasonably sure that we can deliver, and that's a way to gain confidence.
And maybe pushing you a little bit on this part. I mean, on these two aspects, both the immune response and would you see that there are ways of scaling it faster I mean, you didn't use the word of sandboxes, but you touched a lot about the regulatory aspect that sometimes puts hurdles that can be understood from a societal protection point of view, less when it's about repurposing. What could be done better? Is it about this collective intelligence? What we realized, for example, is that having all these teams working together is one thing, and it's great. But then the collective intelligence, as Thomas mentioned, that will come out of list of compounds that will be found by teams from different sides of the world with different methodologies will obviously increase much the chance of success much higher. Therefore, the speed. I mean, what else could be done? Who do we need to have around the table?
Well, probably you need a patients group. I've alluded to it already, but there's a mismatch between the restrictions on the use of patient material for medical research. There's a mismatch between what the patients would like to achieve and what the ethics committees will allow to happen. So I think that's wrong. You can make a lot of a very strong case for great care with the delivery of drugs to patients, because we all get enthusiastic about that. Sometimes the drugs do harm. But in terms of the use of samples, I mean everything from blood to post-mortem material, there is very little chance of doing damage at that point. So it's all upside, but yet we have a huge number of restrictions. And in the case of COVID-19, we don't actually understand how people are dying properly. We don't actually understand to what extent the pathology is direct viral infection of different cells versus immune standby pathology. So that's a fundamental thing that is required to target treatment. Without a large amount of clinical material and the very careful study, we're going to have delays, which are entirely avoidable.
Well, thank you very much. I don't know if Thomas has maybe a concluding question.
Right, there's a couple of questions from the audience. So maybe one I can take here. So it says it seems that different therapies must be applied depending on the state of the COVID-19 disease. Maybe more efforts needed in the differential diagnostics. I think this is also, you already hinted to that point of.
That's really what I said. I mean, differential diagnosis usually means the diagnosis of COVID-19, but I think that question really alludes to the different clinical spectra that you might need different drugs, obviously in the viral phase of the disease and what's now considered to be an immune phase of the disease and the vasculopathy phase of the disease. We need ways of distinguishing those phases and applying different treatments, yes, for sure.
Right, maybe last question also from the audience. They ask, are there also clearly identified scientific and technical challenges to be addressed for emerging treatments on future epidemics? I mean, this is already, you already addressed some of these points, but maybe you can comment on the technical challenges.
On what was that? Just repeat for me.
So you already pointed out that we should have a general preparedness for viral vectors, oligonucleotide interventions, but are there technical challenges, things that maybe techniques that we don't control yet or don't understand yet well enough to actually get to this level of preparedness? So do we need to make new technical inventions that are still missing?
Basically, Sir Peter, what are the other challenges that JEDI should launch in this field?
I think immunity is a real, really major thing. We need to understand how immunity is generated and the best possible way of getting the most robust immunity. It's a quantitative thing, I think. So there's a lot of ground to cover there. Very much the province of academia. Quite possibly would be generalizable to other threats of this type. A really good area for investment, I would say.
Okay.
Okay.
Well, thank you so much for
No, it's a pleasure. I do have another call, so I'll have to leave you, but do wish you well in this great endeavor.
Thank you so much.
Thank you so much.
Okay. Bye-bye.
It was a pleasure.
Bye-bye.
Thank you.
Okay, so with that, let me share again the agenda. So with that, we can quickly go to the updates by the different performers. And the first of that is from Team AI for Science, Professor Dr. Frank Noy from the Freie University of Berlin. Frank, please unmute, and you can also start your video and share your screen whenever you are ready.
Okay, hi, can you hear me?
Yes, perfect.
All right, very well. So thanks, Thomas, André, and everyone for organizing this. Let me see, how do I go to full screen? Here we are. Okay, so we are a team from Berlin, Free University of Berlin, and we are working at the interface of machine learning and the sciences. And so when all of this COVID craziness happened, we basically rushed to following up on a project we had done earlier with a protein that is very similar to one of the target proteins that could be used to inhibit infection, or at least to reduce virus load. And that is this TMPRSS2 or TMPRSS2 protein. It's a serine protease of the host. So the virus necessarily need to interact with TMPRSS2 in order to get its spike proteins activated. And then an interaction with the ACE2 receptor can initiate cell entry. So this is it's a very exposed protein. It's on the host side. So of course, interacting it could potentially have side effects, but also it's very hard for the virus to work around that. So in that sense, it's a very interesting target. It's also used by other coronaviruses and by some influenza strands. So very interesting target. We also have evidence from this paper here by Stefan Pöhlmann and Christian Trosten. That has been now cited, I think, 2,000 times or something like this, that this is essential, TMPRSS2 activation is essential, and that drugs that are on the Japanese market, especially Camostat, but also later shown Nafamostat, which are anticoagulants, may bind to TMPRSS2, inhibit it, and they do inhibit virus entry. So this is where we started. We have worked on other serine proteases like TMPRSS2 before, and for this reason, we felt that we're in a good position to start here. We established another collaboration with Mark Rothenberg, guy in Cincinnati, who helped us to set up a preliminary assay. This is not a very high throughput assay, but it can be used to test drugs, whether they inhibit TMPRSS2. And then we basically did this in vitro test for these two drugs. And so now we want to go to the screening of many drugs, drug candidates for JEDI. And so the difficulty with this is TMPRSS2 unfortunately doesn't have an experimental structure. So the natural starting point would be the X-ray structure or something like this, but we don't have one. But we have done extensive simulations of other serine proteases that have very similar structures. Therefore we felt quite confident starting with a homology model and then doing extensive MD simulations. And so you have to know a protein structure is not a static thing at all. It's very flexible. This is a movie from another protein, not TMPRSS2, generated in John Codera's group. And we do basically similar simulations where we try to extensively sample the conformational space, so milliseconds of simulation sometimes, which is quite long for all atom MD, and then really probe these different conformations. And so this is exactly what we did for TMPRSS2, starting from a homology model. Here's a short movie. We do high throughput MD simulations combined with active learning that basically try to enhance exploration of the state space. generate a lot of data, find the long-lived metastable states or structures, and with some machine learning techniques. And those define basically target structures for us. So with these target structures, now we can start docking. What we did here actually are also quite long simulations of these two drugs that we knew that they were relevant in order to understand their mechanism with which they inhibit or interact with the protein. And I think we are understanding much better now how these drugs work and what could maybe be modified starting from them in order to make them more specific to this protein. But then our drug development pipeline, this is really what we do for JEDI, is we start with initial drug candidates, which include those two and several other proteins which have been reported or assumed somewhere in the literature to perhaps bind to TMPRSS2. Then we look at these databases, select chemically similar drugs as candidates, score them. This is then using our targets that we get from the MD simulations. And right now we just do flexible docking, which is fast, but it's also very dirty, but that is what we can do on a short time scale. But in the next iterations, what we want to do is extensive free energy calculations on folding at home, like do free energy calculations of thousands or tens of thousands of drugs, and then eventually also in vitro scaling, in vitro screening. Then we take the results of these scores, embed them in some latent space that has been learned in order to embed these molecular structures from the entire sync database or other databases. And then on this latent space, we try to do supervised learning of these scores and predict these scores cheaply across the rest of the database, because that's something that's much faster than all of these scoring approaches, even docking. So that can be really scaled to millions or even a billion compounds. And then again, we take the top scorers there, update our track candidate list, score them again with either of these methods, and go in loops until this is converged. So that's the idea, and this is where we are now. I've been asked by several people if we can share TMPRSS2 structures, and we will do that. We haven't done that so far because we are essentially continuously changing things as we go and generating new data as we go. But we definitely will publish TMPRSS2 structure ensembles as soon as it's possible. And yeah, so this is basically the approach based on which we will formulate our JEDI drug candidate list. And we only work on this target. We're not working on other targets at the moment.
That, like you said, that's great actually, if you could share, because we've got, of course, many questions about TMPRSS2, and it seems like a very promising target. So definitely sharing is caring, and like through MolC, for example, they have a very nice repository that exactly. Also we try to point out to the teams.
Yes.
Okay. Thanks so much, Professor Noe. Thank you. In view of time, we'll move on to the second team, that is Team Belarus, and it will be represented by Ivan Bosko. Ivan, please.
Hello.
Go ahead. Yes, hi. We can hear you. We don't see you.
So I have no camera. Sorry.
Okay.
But I will share my screen.
Great.
So, do you see it? Yes, fine. So hello everyone. My name is Ivan, and we are the team from Belarusian Academy of Sciences. Thank you for the opportunity to present our team and tell you what we are engaged in. Our team comprises specialists from various spheres, from biology and chemistry to data science and computer science. Let's talk about our pipeline for modeling used during this initiative. So on the first step, we chose 3 targets and constructed pharmacophore models for every one of them. Then we carried out virtual screening on the basis of these models in various chemical databases using Fermit-Web tool. On the next step, we tested identified compounds with molecular docking using such tools as Quick-Winner2 for docking and scoring procedures, as well as NN-Score2 for scoring only of the docked complexes that we identified. Then we use the results obtained during this step and structures themselves to train the neural network, which is an adversarial autoencoder, to generate more active compounds with good binding affinity. So a few words about this approach. What we do is we extract descriptors or fingerprints from molecules, and we are trying to capitalize on knowing docking free energy estimation to be able to generate molecules with a preset property of good binding energy. For generated compounds, we conduct docking again and throw them into the general pool for each target. So at the last step, we evaluate the best compounds from the previous steps with molecular dynamics tools. So in our case, we use Amber software. Then let's move to the targets. And the third target is the main protease of coronavirus in its free state. So in other words, it's not been binded to any inhibitor. Starting from the protein database, we applied our pipeline to identify 870,000 compounds after virtual screening step and select the best 10,000 of them, finishing with only 5 top chemicals that are presented on the next slide. So here you can see all the values from every independent method we used, such as Vina, NN-Score, and Amber. Then we can move to the second target. It is the same protease, but binded to the inhibitor called X77. So as you can see, we use the same approach as for the first target to find the 5 top compounds. And the results for these compounds are presented in these tables. So you also can see them. And finally, the last target is spike protein subunit 1. The main difference here, obviously, is the number of identified compounds after the virtual screening. It is considerably lower, but we chose the best ones, and you can see them here. We are working on this problem to find more efficient ligands and more in general for this target to overlay them with our methods. So to conclude, I would like to say that regardless of what happens within this initiative and our submission, we do lack of experimental expertise. And since we have a large number of theoretical results for different problems such as leukemia, breast cancer, HIV, and so on and so forth, and we would like to find partners to cooperate with regarding the experimental part. So if you're interested, please contact us, and thank you for the attention.
Okay, thanks you, Ivan, and we hope definitely that we can synthesize some of your top compounds so you can actually really get some experimental validation. Thank you. So again, if you have time, we will move on to the next team. That is the last team for today, and that's the team of Professor Suman Sirimulla from University of Texas. Suman, please go ahead and share your screen.
Okay. Are you guys seeing my screen?
Yep, perfect.
Okay, great. Yeah. So thanks, Thomas, for having me here to give insight of like our current work. So So our effort, so I'm actually working with a couple different people, so a couple different groups, and also a little bit fortunate to have like working with 3 experimental groups with BSL-3 facilities. So I'm actually, so I'm going to share like, okay, our results, like, okay, what are the things that we are doing from our end. But also I actually, we plan to share some of the experimental results that we have, but actually, so we're still not ready with some of that, but actually that will be shared with the group, with the JEDI group pretty soon. So getting into like what we're focused on. So as you all know, the viral replication process. So we are actually one of the our interest is also looking at the different proteins that are involved in the viral replication process. So we're actually looking at several non-structural proteins that are involved in the viral replication process. So we were mainly focused on these 6 different targets, MPRO, RNA-dependent RNA polymerase, and other ones. So well, our main idea, so like we're actually working in different, we're approaching this problem from different ways. So we're doing both structure-based and ligand-based. So for structure-based, so as we are looking at these 6 proteins mainly, so our main effort was, okay, can we do something quickly? So that was one of the driving factor because of the urgency of this issue. So so first thing what we did was we screened these 6 different proteins using using the enamine library. So they have on shelf like 2.1 billion compounds. So first we started screening those, so we did docking using VINA, followed by rescoring using GLIDE and other using GLIDE and DL score. And then we prioritized 10,000 compounds, and then for each protein, and then we further prioritize the compounds using other different filters such as promiscuity patterns and drug metabolism and pharmacokinetic property prediction. And we selected about 100 compounds for each of the protein, and we sent it for testing. So the 600 compounds are being tested, and I actually wanted to show, give those data for you guys. But so we have already, we tested 2 things. One is a toxicity, and the other one is a antiviral activity. So we have the toxicity results, and most of the compounds are that we have are great, but there are a few. So we are actually waiting for the experimental results, which we would get probably in a day or 2. So those are like about 6 different proteins. So initially we screened the enamine database, which is on the shelf compounds. And then now we are screening the real space compounds, which is about 1.2 billion compounds. So these 1.2 billion compounds, these can be delivered by enamine, but they have a turnaround time of 48 weeks. So for the main protease, actually there were several crystal structures that are available with the fragments from the post-era group. So we tried to take advantage of that one. So I just wanted to discuss a little bit more about this main protease. So here there were 70 co-crystal structures that were available, and then there were several with bound to several fragments. So we selected about 11 receptors from them, and then we did the docking using OpenEye hybrid in this case, and then so we found the compounds that overlay the fragments, and then we selected about 5,000 compounds from there. And we also did rescoring of this compounds the compounds that we found to do, and we did a local minimization and induced fit docking. And we selected another top 5,000 compounds. So these are again, like, okay, so this one is, we in this case, we did 2 different methods. And at the end, when we looked at like the overlap, actually, there isn't that much overlap of the compounds. So for every protein that we are looking, like we are actually working with multiple different docking methods or rescoring methods. And then like, okay, we are selecting different compounds and that we are using it for testing. So I just want to show again, so we are also developing machine learning models. So we have data so we're fortunate to have some data from our experimental collaborators and also from collected from some from the public space. So we have activity data and toxic data of these compounds. So we use these compounds and we build machine learning models. So we build machine learning models using 3 different approaches. So one, we use topological pharmacophore features and physical descriptors and fingerprint-based models. So we built 3 different models. We used about 20 different models that are available in the scikit-learn and different features. So for pharmacophore features, we used TP-ATF features from MyChem tools. And for PhysChem descriptor, we used OLSURF from Molecular Discovery and also from the RDKit. And for fingerprint based, so we use about 19 different fingerprints from the RDKit. And we trained and we used the best model, whichever gives us the best model for each one of them. And we built 3 different models. And at the end, what we did was we used a consensus model by combining all these 3. Okay, so here are our results for the machine learning models. So this is descriptors, fingerprints, and topological. And then we have a consensus. Actually, we found out that we also expected that consensus would work a little bit better, and it actually did. So we have 2 different types of models. So one is for activity, and then the other one is for toxicity. So we built both models, and we also made a web portal that's going to be available pretty soon in few days. So where users can just simply submit their SMILE or give a name of the molecule. It will give the activity prediction and toxicity prediction. And also whatever is in our database, if we have tested a compound, if there is a similarity to the query ligand to, if there is any similarity, then that will also show up in the results. So this web portal is coming pretty soon, and the data, all this data that we used will be available pretty soon. Okay. So yeah, those are the acknowledgements. And I just wanted to acknowledge TAC and Boeing at TAC. So we are also using voluntary computing from using Boeing at TAC, similar to folding at home. But TAC has been a very big resource that we have. So so we are fortunate to have these good resources and some funding agencies. So we got a NSF grant recently, and yeah, thank you all.
Great, thanks, Suman. And please, when your web portal comes online, we will be very happy to share it immediately with the teams on our Discord. So thank you so much. Okay.
Welcome.
So,
maybe a quick point, a question from me to Professor Noé, Bosco, and Professor Sirimulla. For each of these results that you will come up, will you be able to simulate or estimate the KDs of the list that you're going to submit? Maybe starting with Professor Noé.
So within the JEDI submission time scale, we'll only basically do that with docking. So yes, there is an estimate for a binding free energy, but I wouldn't rely on that. In the next iterations, we will do free energy calculations, and eventually, hopefully with the JEDI consortium, but we'll also try parallel tracks, an in vitro assay. So they are definitely, but not within the regular JEDI submission which is, I think,
What's
end
the
of
time
next
frame
week.
you would need for this?
I think it will be at least 1 more month, but probably we will actually go in iterations, even in the free energy calculation, because they're much more informative than the docking. So yeah, don't hold your breath for that. I think it's a good strategy to submit the best candidates now, screen them, and in parallel try to improve and whatever comes out of there, screen that later or so. I think we need to hedge our bets and try to do as many things in parallel as we can.
Absolutely. Maybe Ivan, comment on
So you asked whether will we calculate dissociation constants for our top compounds? Yes.
Right.
Yeah. So I can say that we are planning to calculate that because it is like a common metric for our compounds. Yes. So we have it by one method. We planning to convert our results from docking also to dissociation constants, and also when we compute entropy term for dynamic simulations, we also convert it to dissociation constant, I suppose. So
Suman, you want to add something short on this?
Yeah. So our effort is like, okay, quickly move. So I'm hoping, since we are screening billions of compounds, can we get something really safe and very effective? So if we get that, we can just bypass. So we can just bypass doing like a traditional structure-based drug discovery where we get the do a proper lead optimization. So we're trying to see if we can expedite the lead optimization process. So our effort is, if you get something in low single-digit nanomolar concentration, okay, we can quickly jump to the next step. But obviously, we wanted to go retrospect and like, okay, do like much more structure-based and like, okay, do a proper FEP and all that. And but the idea is to how we can expedite.
Very clear. Thank you.
Okay, thank you. So while I introduce Professor Edwards, I'm going to launch another poll, which is, let's say, very related to his topic. So Professor Aled Edwards is the founder and CEO of the Structural Genomics Consortium. It's a public-private partnership of 22 organizations that advances research in human biology and drug discovery. He's a great advocate of open science, and we're very excited to hear your input on this topic. So let me just wait a couple of seconds before I close the vote. Okay, we have some mixed opinions. I will close the poll. So opinions are divided, it seems. So Professor Edwards, maybe you can.
Yeah, thanks. It's pretty exciting. And so what I would try to convince you at the end of this is your challenge is too small. You guys have to think about a way in which you can impact life on the planet much more than the pandemic. The pandemic is a good entree into it. It's politically astute, but you're aiming too small. And I think that what we really want is that all people should have access to affordable medicines, new medicines, whether you live in Malawi or whether you live in Canada like I do. And so the two barriers to why we can't have a more fair society is, first, our system, the system that we've constructed to biomedical research around the world, and second, the economic system about which we view medicines. They're viewed as assets, not as human rights. And once you start with that sort of fundament and you build a system to do that, you get the ideas that patents are essential. But I'm arguing that patents are only essential if you see the world in this fixed way it is now and not in the world that could be. And I use this slide to sort of give you guys a sense of why drug discovery doesn't work and why we're making progress so slowly. So around the world, we spend about $150 billion a year on biomedical research in the public sector. 150 billion is a lot of money. And if you think we spend that every year, And so you wonder, well, why can't we invent medicines, right? It seems like we have a tremendous amount of knowledge and smart folks thinking. I'm going to argue it's the system, and these sets of slides are focused on that. But think of what the system does now. If you think about Parkinson's disease, right? It's a disease that's had massive amount of money. Parkinson described the disease in 1808, and we still do not understand what causes Parkinson's disease. And so, you know, clearly we're biology is a mystery. And so if you take a drug into the clinic for Parkinson's disease and it doesn't work, it's pretty obvious because we actually have no clue how Parkinson's works. We're just taking random shots on goal. Antivirals is easier because you know that enzyme is involved for viral application. For most diseases, it's a lot more challenging. Okay, so here's a graph. Now, if you think about the human genome, it's a really cool way of sort of looking at the philosophy of science. There are 20,000 genes in us, and 1 million years from now, there'll be 20,000 plus or minus 1. Every gene has a name. And so you can use the PubMed and find out how many papers are there on each gene. It's a very simple experiment. So prior to the sequencing of the human genome, I'm assuming you can see my cursor, but if not, I'm looking at the left. Science pre-2000, we just took the protein kinases as a random set, not, you know, a set of genes in the genome. There's 500 of them, so it's about, you know, 2.5% of the genome. And this axis here is just papers. With papers equals money equals people who work on it. So look at all the activity on few, which makes a lot of sense because none of these kinases have been discovered yet. They weren't sequenced yet. They were mysteries in our genome. But if you rank order the kinases in the same way and fast forward to 2017 and say, okay, 17 years later, if you guys are logicians, you should say, whoa, we don't know how Parkinson's work. We don't know how cancer works. Maybe we should start focusing on the, you know, the dark areas of the genome because maybe some of the answers is there. But you know what happened? That didn't happen. So these are only the papers from 2017. And you can see that most of the activity, there are exceptions, that's BRAF, those are PINK1 and stuff, but most of the activity is on the very same proteins that we were studying last millennium. And you guys all know this. If you try and write a grant on this kinase down here and your first section is, we don't know anything about this kinase, I want to find out about it. You're not going to get the grant because we don't reward funding to mysteries of unknowns. We reward funding to narratives, right? This kinase does this, and the mouse jumps around, and you can get a grant. You also don't have any peer reviewers who know anything about the kinases down here. So you guys can really understand. And then if you're a young professor and you write your first grant, you're told to work on something you know. Well, what do you know? What you worked on before. And so it's self-propelling. And then we all, you know, we're all humans. We all like to get together. We form little groups that work on this kinase, and we get together every year or we used to get together every year at conferences and work, and, you know, have the whole GSK3 beta fan club, right? And that's self-reinforcing. So I would argue that as scientists, we're not daring enough, and it's nothing to do with individuals, it's to do with the system. Now you put nationalism on it, okay? And so I live in Canada, and as I say often, it's genetically known that Canadians are smarter than everyone else. And so therefore, if we just look at the papers that Canadians, so you do the same search string, SARC, so if it's in the abstract, keyword, or title, SARC plus 2017 and Canada. So you're only extracting the Canadian papers from this global, and you'd imagine that Canadians are more daring, or not. Right? And if I do it by Harvard, if I do it by Germany, if I do it by Wellcome Trust, whatever and you use, you get the same pattern. And so it shows that we've created around the world a highly redundant system where it's almost bad redundancy, right? If every Canadian scientist got hit by a bus tomorrow, it wouldn't affect the planet one bit. The work is being done everywhere, okay? And so these are opportunities that we need to be called. We will not cure diseases unless we understand what all these bloody proteins do, right? But our system doesn't allow us to do it, all right? So there is a problem that we have to look in the mirror ourselves. And what's the big challenge in biology? Is it making a compound to a viral target that we already know how to drug? Maybe. Is it developing methods that can be used much more broadly, for example, to discover what all these proteins do? I would argue yes. So society is making, getting a quarter, 25 cents for every dollar to invest in biomedical research because of this incredible duplication of effort. And if I was to spend $1, would I spend it on being the 10,000th dollar on this kinase, or would I focus my efforts down there? And I think the answer is obvious, right? If we were to have where the greatest ROI for a small organization like the one I run, we focus on the proteins that no one gives a shit about. Because eventually someone's going to give a shit about it. Obviously, they've been evolved to be on the planet for a reason. And the other barriers are inequitable economic system that we've created for ourselves as a society, where society, as I said, endorses a model where we treat medicines as assets. It evolved that way. It doesn't have to be that way. There's no law of physics that says companies need to invent medicines. It is, and we never question that. And I think we should. I gotta press down here. Nations, they don't fund science, right, to better the world. They fund science to promote their economic interests. And that's part of the narrative that we tell to our governments. We create intellectual property. We create this. We'll contribute to the economic good of the country. And what that does is it put barriers, not scientist to scientist barriers to collaborate, but organization to organization barriers to collaborate. And then finally, universities increasingly treat knowledge not as a public good, but as a financial asset. As Peter was saying before, in the UK, they're busily trying to convince folks that the universities will patent their way out of this Brexit crisis and generate this magic pool of imaginary money that will flow in because they're so smart, and you know how the English are. Anyway, and I was born over there. So what are the solutions? I say, akin, riffing off what Peter says, it's more organized science. Now, in an area like material science, where there's no way to bound the problem, it's hard to organize, right? But where the human genome is, where it's bounded, you can think, oh, in this small area of the research endeavor, there are ways to think about organizing science. The Human Genome Project was one way that it did it. The other way to think is more daring science. I mean, peer review is death by consensus. For anyone who sat on these panels, right, the really daring ones will not get funded. So we need more efforts like JEDI is trying to do that doesn't have to listen to people. There's more open science, right? A lot of the times this redundant research is because we don't know what's going on, right? So if we shared our science as we go, then it would be building on one another, not competing to repeat. I tell my students, often, oh, don't share, you might be scooped. And I tell my students, well, if you have a fear of being scooped, you're redundant. Do some science that no one else is doing, and you have no fear of being scooped. The only time you're going to be scooped is that if you decide to work on something that's so obvious that everyone works on it. Do something different, right? And unfortunately, our incentive system doesn't work that way. We have to be successful in when I was younger and was in the science world, of course, I competed in the area where everyone else did. I won, and therefore I got my credit. But that's a sad way to do science, and it's particularly bad in biomedicine. And then finally, more generosity. Most of us live in countries that are really, really affluent. No matter how much we want more, I think we need to think about being more equitable on the planet. And the COVID thing is truly highlighting the disparities that exist on the planet. And I should remember you, 500,000 people die every year of malaria, right? It's this, the COVID thing, we have 50,000, 500,000 little children die. And sometimes that's just out of our minds, right? Because it doesn't affect us here. So I think we have to think more deeply as rich people. So you guys, I argue, can be the change, right? Because in 50 years, 50, 100 years, 70, I don't know, we will invent drugs in silico. The costs will come down, and we will be able to understand biology more and invent a whole whack of things more. The question is not if this will happen, but when, right? And that's what I think you guys should be working towards. To get there faster, you have to be organized, which you guys are doing. We all have to be more daring. more open, right? You know, there's always a hesitancy, and on more generous and more realistic. I mean, you guys are not making drugs, you're making hits, right? A drug takes 12 years. You have to optimize for a whole bunch of things that are competing. PK, PD, tox, potency, they're all strings pulling in different directions. So, and I think you need think about more experimentalists, I think, because the experiments that you guys do potentially are not the ones I should do, as an example. So that's it. Thank you very much. I have honestly great hopes for your community. It is the future. And the question is, how do we organize to get there faster? And making 1 billion imaginary molecules that no one gives a shit about yet is probably not the way. There are other ways to think about using your humongous brains for good reasons. And as an experimentalist, our organization would be delighted to help you, and not you guys don't need help, but work with you and try and achieve an end that is more, as it were, practical. Because you should read, all of you, The Idea Factory, which is a really interesting book on how the Bell Labs was managed. You know, the 8 Nobel Prize or whatever Bell Labs. They never considered a product done until it was actually used, right? And so a project wasn't done if I did the screen or if I have some putative compounds. That was not success. Success was at the end, and then you knew it worked. And so that's what I would argue, to try and think about it in that way, to think about the end is a target product of a nanomolar compound that inhibits the enzyme for a start, and see if you guys can get there. Okay, I'll stop talking.
Well, thanks a lot, Alan, for some controversial points, I would say, maybe.
Why controversial?
It's not controversial. It's you're highlighting some of the pain points in academia. So I think there's a lot to be said about the points you raised, like more daring science. Also, my PhD advisor told me when I started my own group, he was like, go for something simple, you know, something you can publish easily. And of course I didn't listen, but I guess I ended up okay. But I think it's, like you said, it's much more difficult to actually do that. But I would like to start a little bit in addition to what you have presented. So a lot of things that you advocated in the past years have kind of materialized in the Meds for Kids project, I would say, so the M4K. And you have written about that. So I'm just, can you maybe very briefly tell about this and what has happened in the past years? Because I read a very nice an open letter in 2018. But maybe you can tell us what happened since then.
Yeah, so the idea is that in this particular M4K is that children always get the short shrift. Companies, and I say it doesn't have to be this way, but currently this is how the world works. Companies are in charge of drug discovery. They don't want to make drugs for children, especially because of the risk. And you can understand from the company point of view, it's not, like I say, it's no one's fault. Everyone's acting as they should in the system that we put together. It's the system that's fucked up, right? But anyway, children don't get drugs made. And so how can we as a society create a system? Now, one could just do it all open, but then there'd be so many criticisms from the policy folks that we'd spend all our times talking about public policy and stuff. So in the drug discovery world, there are in fact many forms of intellectual property that protect your market, which is really what a drug discoverer wants, a market protection. The first one is obviously a patent, which is 20 years from the time of disclosure of that clinical molecule. But also you get the data. So in 1983 or 80, Hatch and Waxman said, oh my God, we need more generic competition. And 83, because costs are going up, right? Not then. But anyway, so what they said is we need to be able to let the generics, generic companies used to have to do the entire clinical trial process independently to get a generic drug approved. And they just didn't do that because it cost too much. So the regulators put what they called an accelerated drug approval process, ANDA. approach and say, well, generic copy, you don't need to copy the whole bloody thing. All you need to do is to copy the key points in, let's say, Pfizer's drug application, so that after the patent's over, you can enter the market in 6 months. And then they thought, well, that's not really fair, because what do the generics do? Pfizer spends $1 billion, gets a drug on the market, they'd sue them to try and overturn the patent. And if they overturn the patent, they could enter the market right away. And Pfizer said, oh man, because patents can be overturned. So the government gave an additional way to protect the market to the inventor. They say, hey, you know, Pfizer, you know the data that you have to submit to the EMA or to the FDA to get your drug approved? That's secret to you. For 5 years in America for a small molecule, 10 years in Europe. And the generic company, to be able to get quick approval, has to see those data. So in fact, they just sit there for 10 years, waiting until that regulatory exclusivity period is finished, and then they sue. And the 3 things you can count on in life: death, taxes, and the lawsuit from the generic firm the day your patent, regulatory exclusivity expires. Patents or not, okay? So we thought, well, most people use this regulatory exclusivity to keep people away and to charge high prices. What if we flip it on its head and say, we don't need it? You get this regardless of whether you have a patent. So why don't we create a process that's consistent with open science, and once our drug approved, we will get regulatory exclusivity. And in America, because it's pediatrics and orphan, we'll have 7 years where no one is allowed to sell that drug. In Europe, it's 12 now. Canada, no one cares about Canada, but anyway, it's 8. And so you have ways that in the existing system that you can practice open science, get your drug approved, and still protect your market. Now, for the pediatrics, there is no market, which is kind of like, you know, Alice in Wonderland, right? You're protecting a market that doesn't exist. But anyway, the people will ask, how do you keep competitors at bay? And you say, well, dude, there's no competitors. Who the hell is going to make a drug for a kid? But that's it can be applied to Alzheimer's. It could be applied to Parkinson's. It could be applied to all diseases. So there are ways to do non-redundant sharing science to learn about the biology and make sure you're the one who's allowed to sell the drug, which is what a patent does.
Right. So this is this new chemical entity exclusivity, right?
That's correct.
For small molecules.
That's exactly right.
And it blocks generic competition. But so why, if that is true, why is pharma still so hung up on their patents and their
well,
protections?
2 reasons. First, you don't get your exclusivity until you're on the market, right? Till you get it starts the day your drug is approved, and pharma wants to keep competitors at bay by doing these sort of Marcouche claims of their compounds, like I claim this in the whole universe. But pharma now is trying to patent only the clinical candidate, because if you patent too early, you actually lose billions of dollars at the back end, right? And so the only patent that really counts is the actual drug, and that's the one that's of most value. And it's so, but if you're going to give a business person, you have the right to have 2 forms of protection, a patent and something else. I mean, hell, you know, again, logically, I'd take the both, right?
So if you think about specifically for the JEDI challenge, so we have many teams who have this collective intelligence and you know, like, how can we take things downstream? You know, if you say, well, there's the different forms of protection. So let's say it's not patents. Let's say it's this you know, NCE, right, new chemical entity, the exclusivity. Yeah. Like, you know, who will take then a share of the pie at the end? You know, because that's still, if we want to get companies involved.
Why do you want the companies involved? What law of physics says that you need to have a company involved?
Right. So how would we fund, let's say, taking things downstream where the costs, you know, we are now in the cheap phase, let's say. So when it starts costing hundreds of millions.
We're funding ours from governments, foundations. The only source of funds that we can't get are venture capital, because obviously the venture capital, 3 priorities of a venture capitalist are make money, make money, make money. They don't care about the drug, right? And so as soon as you bring in somebody who wants a return, then you're in the maximize shareholder value world, and then if the prices will be immoral. And so You know, I would argue that there are plenty of sources. NIH has tons of sources of money for drug discovery in the open. I presume Europe will have the same thing. England has, or UK Wellcome will have the same. So I would hope that your commitment to affordability makes one rethink all the things that you guys imagine must be so, because they're constructs of our society. They don't have to be like that, and you guys can change it. Just like your community changed, I mean, the competition community changed the concept of open source in the 90s, and prior to that, it was, you guys are idiots. IBM had the most patents on the planet, right? And that just rocked the world, created the internet so people in rural India can communicate with each other, right? Very wicked cool. I think we need to do the same thing in medicines and start from the high level. Medicine should be a right for everyone on the planet Earth, not just rich whiteys like me, right? What do we need to do to make that happen? And you guys can help. You guys are such an important part of it.
So that's a very good point. And but do you think there's a risk? So I was reading about other initiatives that started out open, but then to take it in the end to the market, sold to pharma that then put ridiculous prices. So like, what's the risk that pharma still, they don't have a good name. So what's the risk that they still abuse, the generosity?
So you're exactly right. Business people will try and find an angle. That's in theory what they should be doing. We have about 7 or 10 lawyers, can't remember exactly, working on exactly that. What would a bad actor do? How can we protect against a bad actor? What would a bad actor do? And currently we're all right, because if pharma needs patents and we put everything in the public domain, they can't patent, right? And so therefore they're going to stay away from that area because it's unpatentable. It's not like patents protect you. I mean, mevestatin and lovastatin, which are one drug that failed and one that made it by one methyl group. It's like a humongous molecule, one methyl group different. It's so easy to get around a patent. So it's not like, oh, I've got a patent, no one can touch. Well, bullshit. M4K started its program from a patented molecule. The Mer-TK company that Stephen Fry's at started their program from a patented molecule and just went in different directions. And so it's not like this early stage magic patent is going to actually protect you if someone wants to riff off what you've done. So it just costs money. And then imagine we wouldn't be able to have this Zoom conversation. We'd all have CDAs in place, right? We wouldn't be able to tell anybody anything.
But do you think, you said that, big universities, well, all universities, but big universities in particular, they see IP as an asset, as a financial asset. And now, you're asking teams to just, share their things for free. So yeah, but would the teams have to internally, fight with their administration or?
No, because I mean, do you guys ever see anybody from the administration come to your labs and look at what you put in the public domain? Right? First of all, you guys are generating no IP at this early stage. There's nothing of any value, but let's pretend it does. No university has ever stopped any faculty member in 30 years from publishing what they want to publish. Never. And so it's a canard, right? About the kind of behavior that we're supposed to behave at, right? And yeah, and it's true. So let me tell you we'll go back to evidence. So in Canada, in 2010, the last time the data were public, Canada as a nation in its universities made $57 million from licensing intellectual property from universities. That's on a $10 billion R&D budget. Okay, so that's rounding error. But to get that 57 million, we spent 55 million on the tech transfer offices and the legals for a net of 2 million on a $10 billion budget, right? And then to protect that valuable 2 million, we put all these MTAs in place. So we execute about 6,000 MTAs in the country, each take about 2 months to execute. So you can quantify the lost opportunity costs, and FTE rates about $250,000 in the industry. So that's $20,000 times this. We're losing $200 million a year in the protecting our IP effort at academic institutions. you guys know the best IP walks on 2 feet. Our job is to train young people who can get out there and do stuff. This new world where we can, as a collective, help make medicines for the world, I think is a wicked cool application of our academic freedom.
But do you believe, so do you think this is relevant only for, let's say, drug discovery in medicine? Or because I know of examples where, you know, some university IP got spun out into nice companies that actually, you know, grow and actually contribute economically.
Yeah, sure. But most 60% of spin out companies from universities don't have any IP, for one. And for two, ask those companies, was your IP important to the ultimate product that you make? And it's an even smaller number. So from the neuro, which we did a case study on the McGill Hospital, they had 15 startup companies, 7 were spun out based on patents, and only 1 says the patent's important. And so if you eliminate patents, you'd still have the 14. You may not have that 1, but on the upside, you think of what else happens if you go open. And what the neuro has seen is a massive increase in donations and in grants because they created a position where we're sharing, you know, to Peter's point, they look after patients. They want them to make cures, right? The patent system hasn't helped us cure Parkinson's in 200 years, right? So maybe it's time to change. As you say, use the collective power of everybody's brains.
So an interesting point also you wrote in this open letter in the Welcome Open Res 2018 is that you say that this open science can increase reproducibility. So
for sure.
Why do you think that is?
So what I have started lots of biotechs, right? So I'm not naive into that world. So when you're doing a drug discovery program in a biotech, your job is to tell everybody the good stuff, right? You need to get that next round of financing. You want to be pseudo open, but if an experiment doesn't work and you're not really sure, you won't tell anybody, right? You'll just say, okay, I'll better keep that secret. And you'll argue, oh, we haven't repeated it, so you know, it's best not to show. When in reality, you don't want to show any dirty laundry, right? And so when you do it openly and you submit your ideas to the community, you will get people phoning in and saying, as they're doing, you know, that compound's got some Michael acceptors, watch out, right? Don't take that one forward. Or I saw that in one of my experience 10 years ago when I was running a program in Glaxo. I would stay away from that functional group, etc. And as a result, then the experiments, you know, there's no hidden, you know, you have your experiment protocols online, you see it. I imagine it's the same as your field, right? If you somebody runs an algorithm and just tells you the results and you don't see actually what they run, you know, you have suspicion. And if they make everything transparent, it's better. There's a terrible thing in science now, biomedical more, to show only your good stuff. Right? You think about how we write papers, write a story. You don't write the science, you create a narrative, right? And if something is inconsistent with your narrative, maybe it doesn't go in your paper, right? Or if you publish submit to Nature and they say you can get it in if you show this. That's a pretty perverse incentive, isn't it? Right? I will get it in if you show that the mouse dies with this compound, right? Well, you damn well make sure the mouse dies, right? And then you get your paper.
I think I mean, we did not choose the word Jedi for nothing. So we are animated by some kind of a rebellion.
Excellent.
I think you are exactly in the same mindset. But I would have a specific question on your very interesting chart you had on the focus on just a few genes. I mean, the universe of the unexplored is obviously much larger. How would you prioritize? Because we all want, I mean, your 2 words were impact and go for the unknown. But so how do you maximize the impact going? Where do you start?
It depends.
Which is, by the way, a question we have when we launch challenges, as we always try to see where's the maximum impact for the effort. So
there are many ways to go into that unknown. Human genetics is one way, right? There are many disease-linked genes in the unknown that are technically difficult to work on. So people will prefer to work on the ones that are known. we're structural biologists and biochemists. A lot of the genes in the genome belong in families that are related by a common structure and common chemistry. And so if you have a family of 450 and 50 are studied, then clearly we could use the same platform to study the remaining 400. And so that's the way that our organization slices and dices the unknown. I'm sure up to the genius of other scientists around the world who have different methods, they could do it differently. What we have noticed, though, to your community is the most powerful way to get people moving into the unknown is to make a tool compound available, a high quality chemical inhibitor. Because from an experimentalist, it's the simplest reagent. It's cheap to buy, you squirt it on your cells, and you can see what's happening. And so of all the experimental reagents that will convince an experimentalist to go into the unknown, a chemical tool is number one. And you saw some of the downstream peaks. Remember there was this peak, but there were some big peaks. A lot of those BRAF, it has a drug against it. So people would squirt things, it's really easy for people to do. So we're proactively creating these free chemical tools to try and induce people into the unknown. And, probably 70 clinical trials are ongoing because of a new target that we validated in quotation marks with a chemical tool that we made freely available. So it's an interesting strategy, maybe aligned with this kind of stuff that you guys on the Zoom here can do. But it only is useful if it's freely available. It's hiding in your IP and you make everyone sign away their firstborn before they have that compound. Your work's going to go and hide in the bowels of history.
I think the message is loud and clear. Thomas, you have a few.
Yeah, so a question that was raised a bit earlier in the Q&A through the system was, how, and it feeds back to the first discussion with Sir Ratcliffe, like how can we make theory groups, experimental groups, biomedical groups all work together more coherently instead of pursuing small papers and academic kudos.
I think you have to find a Venn diagram where everyone's interests are aligned. And so in our organization, the first mechanism to do that is you declare open. So we have 10 pharmaceutical companies inside the SGC who share their stuff. Now, they don't share everything they do, but in the box with us, it's shared. And then you start to think about, again, to the idea factory, what do you guys actually, what's success for your first phase of your JEDI? Is it a nanomolar compound that inhibits of a new chemotype? Is it a drug? Is it, and I think bite-sized realistic steps towards the end, and the end is complete in silico drug discovery. And that will happen, right? But I don't think you can do what you're doing now and say you're inventing a drug, right? We wouldn't even put a compound on cells until we know it's nanomolar binder to the protein, has a nice KD, has an off rate, the SAR, like if you change the structure of the compound, the structure, the activity against the protein changes. Only then would we think even close to talking to a biologist. Because a low affinity compound is so pleiotropic, it'll do all sorts of shit. And if you're only looking at antiviral, you go, oh, it's antiviral. Yeah, well, it's antiviral and it's an anti-Golgi, but you're only looking through the antiviral lens, right? So you won't get specific until you're in the double-digit nanomolar, right? And so you have to, anyway, that's the kind of practical stuff that we could do together because we have a lot of experience on that side.
Right. I mean, the metric of success for stage 1 is a better than 100 nanomolar binder, and we cross our fingers that we'll get it. But
binder, that's a great one. Binder biochemically.
Yeah, exactly. Yeah. So and we hope but we're trying to think further down the line. Like we don't want to just have a nice binder. We want to take it all the way.
I don't know how many computational guys I've collaborated with in the last 15 years. You know how many compounds have come out in the 15 years? So if you guys can find 100 nanomolar binder, I will be singing from the rooftops how cool you guys are, right? It's not easy what you guys are doing. The binders you're getting, if you do from a serine protease, there's so much chemistry to build off of. It's not a naive target, right? You guys are not cheating, but you're using a lot of experimental data to guide your thinking. If I gave you a virgin target and you found me an animal or binder, I'd vote you for God.
I think that's the biggest motivation call for all the 30 plus performers that are on this call. Maybe, I mean, we have already stretched over 15 minutes of the scheduled time. Do you have a last question, Thomas, before I give a few words and then you conclude?
I think you can wrap up.
All right. Just maybe you will give us a few takeaways. But first, I want to give a few thank yous. First to Aled Edwards. So thanks for this very straightforward and fresh intervention. Peter Ratcliffe has been great too. Thank you to the three presentations which were made, and I want also to give a thank you to the foundations that are also supporting our work, especially the AXA Research Fund, who has been instrumental in this adventure. The second thing is a little bit housekeeping. We know you are very focused on the screening and on the modeling and on the docking for those who use these methodologies, but keep us posted by letting us know where you are, so it allows us to understand where the different teams are. Follow our Twitter account, EuroJEDI. And for those who are also going beyond this COVID, I also want to make sure that you know that JEDI is tackling challenges in many areas, in the environmental field, in the healthcare field, obviously, but also in the digital field. For example, I saw one of the teams presenting GANs. We will soon launch a challenge on deepfakes, which is obviously another of these big societal challenges. And last, I mentioned during the discussion with Peter Ratcliffe that we will launch this pandemic preparedness initiative with two pillars. One is focus on the unknown, focus on those generic discoveries that could have big impact. So we'll definitely come back to you, and Aled, you are part of it. I mean, there's no way out with everything you said now. And the other thing is we think about some kind of a task force that could bring together a few key scientists, but not the traditional scientific committees that are brought in by policymakers when they need advice on confinement and so on, but really something which can be a sparring partner, and that could also go international. Obviously, this is on cross borders. This is a huge endeavor, but we think that building on what we have launched with the challenge, we can do it. So this is all for me. Thank you to all, and please, Thomas, share with us what you got out of this very exciting.
Right, so I think some key lessons that we could extract from today's webinar is this, finding this balance between keeping science open but still keeping interest of all partners, academic, industrial, and regulatory, so the governments. I think definitely it's a good idea to have some smart scientists in governments. I'm not running, but okay. I think that definitely improves things if there's an understanding between the political side and the scientific side. And I really like this message of seeing medicine as a fundamental right, not as a way to improve your return on investment, which seems to be still the case at the moment. So I think we just have to find the right way to build a framework around that actually brings people together in a constructive way, because that's a bit what's missing at the moment, I think. So definitely we will need more discussions to come up with some nice ideas for that. So I think with that, I would like to wrap up this session. So André, you're muted, sorry.
I just wanted to say also thank you to you because you've been instrumental in this. So you're really running the show here. Okay, so we're planning to do another webinar. We will let you know as soon as possible with the dates, and we hope that you are still enthusiastic enough to join our endeavors, and hope to see you next time. Thanks a lot. Bye, everyone.